All right. Hi, everyone. So my name's Kristy. I'm currently a PhD candidate at UQ in the field of plant ecology. So it's my title slide. And the title of my talk today is Impacts of Myrtle Rust-Induced Tree Mortality on Nutrient Cycling in a Wet Sclerophyll Forest. I'd like to begin today by acknowledging the traditional owners of the lands on which this research was conducted, the Yagambeh, Yagara and Turrbal peoples. I'd like to pay my respects to their elders, past, present and emerging. So over the last 2 days, we've heard a lot about the fact that myrtle rust has been causing these significant declines in populations of several highly susceptible species since it became established in Australia in 2010. But what we know much less about is how the loss of these individuals is affecting the dynamics of forest communities. What functional traits or roles did these species have, and how might their loss affect the functioning of a forest in the short and longer term? These are questions we know much less about and that have formed the focus of my PhD research. In forest communities, ecosystem functions are driven by functional traits. So any trait which impacts fitness indirectly via its effects on growth, reproduction and survival. So for example, leaf traits such as leaf dry matter content or nitrogen content influence nutrient cycling, so the cycling of nitrogen and carbon in forests, while dispersal and reproductive traits such as fruit size or seed dispersal mechanism influence cross-trophic level interactions such as bird-tree interactions. These were two of the functions that I looked at in my research. So my study site's probably quite familiar to a lot of you. Jeff mentioned it on the first day and Tilly spoke again about it today. It's the Tullabudgera Valley site, so it's in the Gold Coast hinterland, predominantly wet sclerophyll forest, so it's eucalypt and brush box canopy with a rainforest understory. It's also predominantly regrowth forest, so recovery after past land clearance, and it's experienced really high tree mortality in the midstory and understory of the forest. So you can see my plots up there in the centre diagram. So I had 32 circular plots with the 2 metre radius located across the site, very close to where Jeff Pegg set up the long-term monitoring transects. And just acknowledging here, they did represent slightly different microclimates. I collected my data over a La Nina period, so between 2020 and 2022, that followed a very strong El Nino period in 2019 that saw some of the driest days on record on the east coast of Australia. I used several estimators of disease impact. So one was based on basal area. So I measured the basal area of all the plots dead and alive, all the trees in my plots, and then divided them to get one metric. You can see here from this figure that they are positively correlated with this one exception of this point out the side, which is a plot that has a very large Eucalyptus grandis with a DBH of 68, which was nearly double the size of any of the other trees and considerably higher than the median of about 6. This metric varied between my plots, so from 100% tree mortality to 0%, which meant all the trees were alive, but there was at least one myrtaceae in that plot that was at varying stages of being alive. And it differed significantly, as I said, between the plots, but the median didn't differ between the microhabitats that I mentioned. So I focused on nutrient cycling. I'm not the first one to look at this. These are a couple of examples from the US. So they looked at beech bark disease and Phytophthora ramorum and found that in plots of increasing levels of mortality, they saw impacts on carbon and nitrogen content of litterfall, nitrate, ammonium, and soil carbon to nitrogen ratio, and suggested the strongest driver was as a result of changes in species composition. So the new species that were coming in differed in their litterfall, the content of nitrogen and carbon. And another couple of differences between these studies and mine is that the pathogen had been established in these plots for a much longer period, and the forests were a lot simpler than the forest that I was working in. So here are some of my results. So we've seen these kind of figures in other presentations. So it's kind of like a map. The points on the plot represent each one of the plots that I had. And you can see there's some overlap between the different microhabitats, indicating the variability in the factors was quite similar within these sites. You can also see the PC1. The strongest drivers of the variation were the soil texture components, so clay and sand, which were inversely related, and PC2, the strongest contributors were temperature and foliar lithophore, which were positively correlated. But most importantly, PBA, which is my measure of disease impact, how much variability was that contributing? For PC1 and PC2, very little, but for PC3, it was a strong contributor of variance, which is, pretty notable given the complexity of this community and the comparatively short period that the pathogen's been there. I also looked a bit at carbon stores, so plugged the DBH of all the trees into some allometric equations and from then estimated carbon. So around 21% of the carbon in my stored plots was located in dead trees. This plays out to about 8,000 kilograms per hectare of carbon in dead trees. You can see from the photo there's a pretty high number of dead trees there. So this could affect the storage of carbon at this site and in the region generally, as these trees are no longer actively photosynthesizing, and as they fall and begin to be decomposed, they could affect respiration rates as well. And as Tilly highlighted so well in her talk earlier, just the risk that this could create to fire as well. Wet sclerophyll forests need fire. The structure and composition of these forests is a result of repeated burning by First Nations people over thousands of years. But the intensity and the frequency of fire matters. And the fact that there's so much fuel and the structure of the fuel could increase the risk of severe fire, particularly as we move into an El Nino period. I also wanted to touch on some of my results from a paper that we recently published on the first chapter of my PhD. So in this paper, we looked at some dispersal and reproductive traits, so making a comparison between the myrtaceae trees that were previously dominant in the mid-story and understory and the seedling community, which will eventually grow up and replace these trees. So we looked at seed dispersal mechanism. So positively, looks like we're not going to see a shift away from bird dispersal, which is good. But in the short term, as Tilly also mentioned, we're likely to see a reduction in fruit availability as so many of these trees have died. They're no longer actively producing fruit, and it will take several years for those seedlings to grow up and produce fruit. And also potential shift in dominant fruit type. So a lot of these myrtaceae, like the one pictured here, produce berries, while a lot of the seedlings that are coming up produce drupes, so kind of like a cherry with a single seed. Birds have different preferences for the fruits they like, particularly a lot of the seedlings that are coming up are from the Laraceae family, which some of them invest in really high nutritious pulp, which some true frugivores prefer over these berries. So we could see a shift in bird visitation patterns in the longer term. Another result is possible loss of this key functional group. So species such as Archaerotomyrtus beckleri and Rhodamnia rubescens were likely key early colonisers at this site. So I mentioned it was previously cleared, so helping to recapture. But with the dramatic declines and potential loss of these species, the question remains, which species will then fill this role? I observed both native and non-native species that like these disturbed kind of conditions. So if another disturbance happens, monitoring will be needed to see which one fills that role or which species. And the final is just the impact of climate, particularly rainfall, at this site. So as I mentioned, my study was conducted in a La Nina period, but the long-term monitoring at this site allowed the observation of just increased recruitment across the site with the rainfall. Big flush of growth. Rainfall trumps most things in this system, it looks like. So encouragingly, 90% of the seedlings I recorded were native, so that's good. And 14 of the 15 species were associated with this local regional ecosystem, which is also good. But considering how dominant these myrtaceae were, the fact that I observed so few seedlings really indicates that they weren't in the seed bank anymore, or just that myrtle rust was killing them as they germinated. So a big impact still on the myrtaceae. In conclusion, myrtle rust has significantly affected the health of several highly susceptible species in this forest, and it appears to be affecting some ecosystem properties and functions, including cross-trophic level interactions, resilience to future disturbance events, carbon storage, fuel loads, and to a lesser extent, nutrient flux. And favourable rainfall conditions really appeared to be aiding the recovery of this forest, but with few individuals from the myrtaceae species that were previously really dominant. And a big thank you particularly to the landholders who've hosted us at the site for so many years. the funders behind this research, all the volunteers who gave me a hand in the field, and the experts who offered advice through this process. And if anyone has any questions.
That was a really wonderful presentation, thank you. And it's really great to hear some of the answers to those questions about how ecosystems might recover from myrtle rust. But I'm just curious, it's a bit of a tangential question, but in any of your reading for doing this research, have you seen anything quantifying the impacts that these diseases like myrtle rust and other ecological plant diseases have on carbon storage?
Yeah, there's a bunch of research out of the US. So I acknowledge there's a lot of factors that I didn't measure. So they went much further into looking at respiration rates and also recruitment has a big impact. You know, if we're having heaps of things growing, maybe they're sequestering more carbon than these trees that were dying off. So a lot more research would be really needed to determine it. But I could send you the citations, but I feel like, Phytophthora ramorum, they've done a heap of work on ecological impacts of that pathogen. Also on fuel loads, they've done a heap of work. So I feel like there's a lot we can, be great to reach out to them and see if they have any tips in this space on how we could look at myrtle rust as well. Yeah.
Any more questions? I had a kind of similar one, I suppose. This is kind of one site in Australia that's really being monitored detail.
Yeah.
But I was wondering about the carbon question and going wider. Do we have we quantified the impact the myrtle rust is having in Australia to the point where we could actually look at how much carbon we're losing due to it?
That would be a great thing. If someone wants to answer that question, we should definitely get someone on that. No, I've just done it at this site. Yeah.
Can we extrapolate at all? Or?
I mean, the forests just differ so much. And I think, also I've heard the variability and susceptibility. So the Melaleuca, you're only losing a portion of the trees. Mm-hmm. Maybe you'd have a lesser effect there than you would where you're losing just a high number of trees. So maybe difficult to extrapolate beyond this region for now. Yeah. But someone should definitely do that if anyone wants to. Yeah.
Cool. Any more questions?